Another part is about lipids. How do we define lipids? Lipids are intracellular compounds which are insoluble or poorly soluble in water. They are readily soluble in non-polar solvents such as ether, chloroform or benzene. The hydrophobic nature of lipid is due to the predominance of hydrocarbons in their structure. Where do lipids comes from? Lipids are molecules that can be extracted from plants and animals using non-polar solvents such as ether, chloroform and acetone. Fat belongs to this group, as do other steroids. Lipids has various functions to perform in our body. They serve as a storage form of metabolic fuels. They also serve as a transport form of metabolic fuels. Lipids provide structural components of membrane. They also have a protective functions in bacteria, plants, insects and vertebrates. There it serves as a part of outer coating between the body of organisms and the environment. How do we classify lipids? There are various ways of classification of lipid, but one of the most common way is lipids are recognized or classified in 5 different ways. First, fatty acids and their immediate derivatives. For example, prostaglandins. Second, glycerol esters. For example, acyl glycerols or phosphoglycerides. Third, sphingolipids. Example, sphingomyelin, glycosphingolipids. Fourth, cholesterol and its derivatives. For example, esters of cholesterol. And the last one is isoprene derivatives. Various vitamins such as A, vitamin E and vitamin K are the example of this. Now we will discuss some examples of lipids in detail. First one, fatty acids. What is fatty acids? They are the building blocks of neutral fat molecules. There are 2 types of fatty acids, saturated fatty acids and unsaturated fatty acids. Saturated fatty acids are those which have a single bond in the carbon. Unsaturated fatty acids are with double bond. All the fatty acids chain differs in the length of chain and have an even and odd number of carbon. Saturated fatty acids have single bonds between the carbon atoms. They are formed from animal sources and are solid at room temperature. Lauric acid, myristic acid, palmitic acid, stearic acid and archidaic acid are the examples of saturated fatty acids. Fatty acids with double bond are called as unsaturated fatty acids. Fatty acids with one double bond are monosaturated, while those with 2 or more double bonds are polysaturated fatty acids. Unsaturated fatty acids originate from plants, are liquid at room temperature. Palmitoleic acid, oleic acid, linoleic acid, linolenic acids and arachidonic acids are few examples of unsaturated fatty acids. Saturated fatty acids and unsaturated fatty acids differs in their melting points. The higher the melting points of the saturated fatty acids reveal the uniform rod-like shape of their molecules. The cis double bonds in the unsaturated fatty acid leads a twist in their shape, which makes it more difficult to pack their molecules together in a stable repeating array or crystalline lattice. 2 polyunsaturated fatty acids like linoleic and linolenic are considered as essential because there are absence in human diet, which leads to the health problems such as scaly skin, stunted growth, and increased dehydration. These acids are also precursors to the prostaglandins, which is a family of physiologically effective lipids present in minute amount in most body tissues. Another examples of lipids, fats and oil. The triesters of fatty acids with glycerol make up the class of lipids known as fats and oils. These triglycerides are found in both plants and animals. Breads and pastries from a bread factory often contain mono and diglycerides as dough conditioners. Diglycerides that are solid or semi-solid at room temperature are classified as fats, and those triglycerides that are liquid are called as oils. Fat occurs predominantly in animals and oil originates chiefly in plants. Properties of fat shows the predominance of saturated fatty acids and oils composed of unsaturated fatty acids. The difference between oils and fat lies in their melting temperatures rather than fundamental structural difference. The constituent compounds includes tetradecanoic acid, hexadecanoic acid, linoleic acid, oleic acid and glycerol. Another example of lipid is wax. Waxes are esters of fatty acids with long-chain monohydric alcohols. Natural waxes are often combinations of such esters and may also contain hydrocarbons. Waxes are widely distributed in nature. Waxes form a protective covering over plants and fruits from dehydration and small predators. Feathers of birds and fur of some animals have similar coating which serve as a water repellent. Carnauba wax is valued for its toughness and water resistance. Another common example of lipid is phospholipids. Phospholipids are complex fat derivatives in which one fatty acid has been replaced by a phosphate group and one of several nitrogen-containing molecules. Phospholipids form the largest class of lipids in all biological membranes. All phospholipid molecules are amphipathic in nature. This property makes two layers of phospholipid molecules in all biological membranes, which spread as sheets. The polar heads face towards the outer and the cytoplasmic side, while the non-polar tails are present in the inner side, forming the hydrophobic core of the membrane. Because of this property of biological membrane, it does not allow polar molecules and ions to pass through them with relative ease. Some phospholipids from unicellular organisms and vertebrate heart tissues contain ether linkages instead of ester linkages. For example, plasmalogon, platelet activating factor, is another such phospholipid in vertebrates. It is released from basophils, which stimulates platelet aggregation and release of serotonin. There are two major classes of phospholipids. One, glycerophospholipids. Glycerophospholipid contain glycerol as alcohol. Another one, sphingophospholipid, which contain sphingosine as the alcohol. Glycerophospholipids are the main lipids that occurs in biological membrane. It consists of glycerol 3-phosphate esterified at its carbon 1 and carbon 2 with fatty acids. Generally, carbon 1 contains saturated fatty acids, whereas carbon 2 contains unsaturated fatty acids. There are different types of glycerophospholipids. For example, the most common one, phosphatidic acid. What is phosphatidic acid? is the simplest phospholipid. It is the intermediate in the synthesis of triacyl glycerols and phospholipids. It is present in low concentration in tissues. Another type of glycerophospholipid is phosphatidylcholine. It is also called as lecithins. This class are the most abundant group in phospholipid in cell membrane. Chemically, it is phosphatidic acid with choline as the base. It represents the storage form of body's choline. Another type is phosphoethanolamine, also called as cephalins. It is the nitrogenous base present in the cephalins. Another type is phosphatidylinositol. Phosphatidylinositol is the stereoisomer myinositol is attached to the phosphatidic acid to give phosphatidylinositol. Another type is phosphatidylserine, which is named as amino acid serine present in this group. Another example is plasmalogens, which is when a fatty acid is attached by a ether linkage at carbon 1 of glycerol in the glycerophospholipid. The last type is cardiolipin. Its name as it was first isolated from the heart muscles. Chemically, it consists of two molecules of phosphatidic acid held by an additional glycerol through phosphate groups. Another type is sphingolipids. Sphingolipid is the second largest class of membrane lipids. Sphingolipids, like phospholipids, are amphipathic, having a polar head and two non-polar tails. They do not have glycerol in their backbone. All sphingolipids are derived from a compound called as sphingosine. A molecule of phospholipid is attached to the amino group in an amide linkage at the carbon 2, which results in ceramide. Ceramide is the parent compound for all sphingolipids. A polar head group is attached to the first carbon atom by a phosphodiester or glycosidic bond. This results in an amphipathic molecules. There are three groups of sphingolipids: sphingomyelin, glycosphingolipids, and gangliosides. Sphingomyelins is a structure in which polar head group is phosphocholine or phosphoethanolamine. It is present in the plasma membrane of all animal cells and myelin. Another class, glycosphingolipids. These are present on the outer surface of plasma membrane. The polar head groups are one or more monosaccharides attached to the hydroxyl group at the carbon of ceramide. Galactose is the head group in the plasma membrane of neural tissue, while glucose in the non-neural tissue. Glycosphingolipids acts as a cell surface recognition molecules, especially as blood group substances. Another class is gangliosides. Gangliosides are complex sphingolipids with oligosaccharide as head, having sialic acid at the termini. Sialic acid conforms a negative charge to the sphingolipids at pH 7. Sialic acid confers a negative charge to the sphingolipid at pH 7. Another class of lipid is steroids. Steroids are lipids that contain 4 carbon rings joined to form a steroid nucleus. Steroids are oxidized derivative of sterols. All steroid hormones are derived from the sterol, cholesterol, a 27 carbon compound, and 8 carbon side chain is attached to the steroid nucleus, which result in a cholesterol molecule. Cholic acid are the metabolic product of cholesterol formed in the liver. These are released to the gallbladder where they form bile salts. Vitamin D, a fat-soluble vitamin, is derived from the sterol. And the last class of lipid is lipid-soluble vitamins. Vitamins are organic compounds that human tissues cannot synthesize, but they are required for normal growth and development. Vitamins are classified as water-soluble or fat-soluble. Water-soluble vitamins such as vitamin C, which are rapidly eliminated from the body and their dietary levels need to be relatively high. The lipid-soluble vitamins are not as easily eliminated and may accumulate to toxic levels if consumed in large quantity. Fat-soluble vitamins are vitamin A, vitamin D, vitamin E, and vitamin K. Fat-soluble vitamins can be regarded as being built up from isoprene units. Human tissues can manufacture vitamin D, but not the other fat-soluble vitamins, although they can make and handle isoprene units in the synthesis of cholesterol and ubiquinone. Human tissues can manufacture vitamin D, but not the other fat-soluble vitamins, although they can make and handle isoprene units in the synthesis of cholesterol and ubiquinone. With this, we end with the basics of carbohydrates and lipids. Thank you.